Good afternoon, ladies and gentlemen. A very warm welcome from the Nano Safety Cluster, Working Group A on Education, Training and Communication, and also from the NanoCommons project. We welcome you to our today's webinar on molecular initiating events prediction on the tool. So we have with us Abhijit Dasgupta from the University College Dublin in Ireland, who is going to give us a webinar on the molecular initiating events prediction tool. We also have further with us two more experts on this field, so Vladimir Lobashkin, also from the University College Dublin, and Vadim Tsernovkov, also from there. And this team is at your availability to give you insight in this prediction tool, which is an integral part of the NanoCommons knowledge infrastructure. I will, for the first couple of minutes, just give you a little bit of background and context of our today's topic, which falls into the area of adverse outcome pathways, and we'll also give you some insight on related topics that are already training materials available online. I'm Martin Himli from the University of Salzburg, and I'm chairing the Working Group A. So this is a joint education event of the Nano Safety Cluster, Working Group A, and the NanoCommons project. I see people are popping in, and I guess that we're gonna start with our webinar today. I would like to highlight once again, make you aware we are recording this, and to my knowledge we have obtained your permission by registration. I hope this is fine. You're very welcome to ask questions at any time. Please make use of that. You have at your panel the chat box. Type your question at any time into the chat. We are going to have Abhijit having the presentation of the prediction tool, and then afterwards, during the demo, he will be available to answer questions at any time. So just to give a little bit of background, what NanoCommons is about, we are an infrastructure project of the European Commission funded theme, and this graphic basically highlights our intention. So we are also understanding ourselves as a networking activity. We are a community building infrastructure, and you see here by these symbols, there's logos, many different also databases and tools, resources, which we try to link together in order to fill the gap of data, fragmented data that still exists in the field of nanosafety assessment. How do we do that? We are offering a number of services and tools and models. We try to make them interoperable, and we do this by kind of gathering them around the entire life cycle of data. So if you look at the left side, when starting for kind of generating data, we are supporting experimental workflows, the design and implementation of nanosafety experiments, for example. When data needs to be processed, analyzed, we are offering a number of tools in this regard in the other category. And even data visualization, predictive toxicity, model building, things like that are included in the third category. And still, also in the fourth category, we are supporting data storage and make data interoperable with each other. So try to link different resources. While the project is still ongoing, you can, and you are very invited to do that, apply for transnational access to also get support in using some of these tools. And even also there is an open TA call still for getting your tool, your model, if you're a model builder, integrated into our infrastructure to make it eventually enrich and get a wider application of your model. So please consult us. Here is the email address, and get in touch with our experts. We will then kind of direct this into the correct direction where people can then kind of synthesize this TA application together with you. So what we do have in terms of education resources is also a user guidance handbook, which you find under this internet link, which is displayed here. That also gives you a nice compilation of different online available tools, which we have already established, training materials, aligned with this data lifecycle categories that I have highlighted earlier. What we do have, for example, is also our events log, and here you see highlighted the event of today, for example. If you get there, you would also find easy access to resources that are linked to this theme of the event, for example, for today. And there is a second one which are highlighted here. We had already some other educational events on AOP related topics just 2 months ago, for example. There are further events that went into this direction. If we scroll further down in our events log, for example, we had last fall the Nano Safety Cluster Education Day, and there was an entire session. The session 1 dealt with AOP and systems biology approaches to nanotoxicity testing, nano safety assessment. I would also like to highlight here there was this Smart Nanotox International online conference, the final conference, where there were resources also, and there are recordings available. Go to this link. You would then be kind of shortcut to have access to recordings that give you eventually some nice background on the topic of today, or maybe even kind of guide you further to other related topics regarding adverse outcome pathway. You do also find our events and our training materials via the Elixir test, so kind of getting us a little bit more into the biosciences area with this, where there's lots of training events and materials established. Highlighted here, once again, 2 events that we had already, and I was introducing them just earlier. The materials are open access available. The slides will be from this webinar today to be uploaded, freely available to Zenodo, and can be downloaded from there. This will be done after this webinar, certainly in a compiled manner. So our activities are in line with also the different working groups that are in the Nano Safety Cluster. And you see here in this circle the different working groups that the European Union Nano Safety Cluster has. For example, materials and standards is working group B, and working group C has different 4 different subgroups basically. And I highlight here the working group D, which is dealing with models and tools. And we had an entire session, and all these recorded materials are available. That were a couple of tools were highlighted for toxicity testing and for toxicity integral workflows, actually. So you find this at the Nano Safety Cluster YouTube channel. All these recordings lie there, and the shortcuts I have shown earlier to have easy access there. Another working group, the working group F, is dealing with data management. We had also a session there at the Nano Safety Cluster Education Day past fall, and this leads me basically to the very important topic that NanoCommons is, the Nano Safety Cluster is very convinced about, is data fairness. So to make data findable, accessible, interoperable, and reusable. So this theme is a very strong focus of our activities in the project and as well in the safety cluster, to make data be able to be reused by other people and enrich data sets that have been provided, generated by experimentalists eventually through in silico workflows. So we try to link in vitro or in vivo workflows with in silico prediction workflows. And this is a big activity in our project. One source how you can link things is actually also the NanoCommons knowledge base, where of course data are a repository. It's a data repository, but through this database, you should also have access to certain sets of tools that are becoming more and more available throughout this project's lifetime. So this shall be for the future a good access point for linking data with tools in order to make things interoperable. So I'm getting close to the end of my introductory presentation. I would just like to highlight what's next, basically. So I would like to announce the nanosafety class, nanosafety training school, which was usually happening physically in Venice. So it's also often regarded to the Venice Nano School. But however, very sadly this year it has again, people had to decide to make it virtual as so many other events. So it's going to be taking place virtually between June 21st and June 25th. I would like to emphasize that it's a very important and interesting event, especially for early stage researchers, so for PhD students, postdocs, to present the results, to discuss with each other, but it's also a quite nice educational program there. And you see here the topics. They actually are also gathering a little bit along with this data life cycle. So from generating data on hazard and exposure, also modeling of exposure, assessment of exposure, going to the field of nanomedical applications even, then modeling, of course, as I have mentioned already today, and getting then a little bit further to regulation. What do we do with this kind of data? So grouping and read across approaches and risk governance are also going to be on display during this event. So here, final slide, a few links that I have mentioned. Once again, the user guidance handbook, the events in our NanoCommons infrastructure, the Elixir tests shortcut. You will find this also on the PDF at the Zenodo then, so you can easily click there and find access to the materials. The YouTube channel, and if you're interested in our education program, please stay tuned with us. Get on to the mailing list of the Working Group A. We are having regular webinars, more interactive training sessions even. Stay in touch with us. There is the internet address below here, and you can subscribe to the mailing list. We're not bothering you a lot. Usually it's just few messages that get through. So this should be not too wasteful in time for anybody. So let's have Abhijit now, and I wish you good success, and hopefully it's a very interesting webinar for your experimental workflows that you can make use of that.
Good afternoon, everybody. I am Abhijit Dasgupta. Next 45 minutes, I will elaborate different aspects of our D5.7 tool, which is a first tool for molecular initiating events or MIE prediction integrated to NanoCommons knowledge base. Before going into main topic, let me introduce some basic terminologies very briefly. The first one is key events, which is an important molecular chemical interaction initiating another molecular incidents. And the second one is molecular initiating events or MIE, which is a special kind of key event which can be considered the first step in an adverse outcome pathway. Here, adverse outcome pathway refers to an unintended or unwanted event or state occurring during or following medical care. Thus, adverse outcome pathway or AOP is a simplified depiction of complex toxicological process in a linear and modular format, starting with the MIE and ending with an adverse outcome. The interaction among multiple AOPs form networks of AOP. And finally, omics is a collection of data about genes, proteins, transcriptions, and metabolites, among others. And this figure clearly illustrates the different utilities of omics in systems biology, as omics carry information of genomics, transcriptomics, proteomics, metabolomics, phenomics, and genomics of a particular species. This figure illustrates how different key events are interacting and originated from a MIE and finally generates an adverse outcome. Such series of interaction forms an adverse outcome pathway, linking molecular and cellular perturbation to impacts at the tissue, organ, organism, and population levels, and the types of assays that can be used to explore the perturbations at the different levels. Hey, in this slide, the first figure is discussed already. I have discussed in the previous slide. I want to mention just one thing, that key events can be measured from different bioassays also. And the second figure shows that not a single MIE can initiate the AOP, but multiple MIEs can be responsible for multiple AOs or adverse outcomes. And I have put this figure just to show how multiple AOPs can interact among them and to form a network of AOP. Now, I want to mention the motivation behind developing the MIE tool. Here, some key points are to be noted. Increasing number of nanomaterials and their various properties lead to the requirement of efficient toxicity testing procedure. The existing animal relevant toxicity testing tools are costly and time consuming. And there is need of alternatives that should be faster, cheaper, and sensitive. The alternative strategy should justify the strategic prioritization of testing and targeting the most apparent outcome. The alternative may be the adverse outcome pathway framework, which provides a pragmatic insights to promote the development of alternative testing strategies. And omics can be used for identifying and describing MIEs and key events for development of quantitative adverse outcomes pathways. However, there are many challenges to overcome. First of all, to understand an MIE completely, a lot of information is required, such as information about chemicals associated with the MIEs, structural features or properties of the chemicals, types of interaction between the chemicals and biomolecule or biosystems, and the nature or structure of the entity with which the molecule interacts. Next, obtaining all such aforementioned information is very much difficult. Thirdly, the most well-studied chemicals lack detailed reports of molecular interaction. Fourthly, nanomaterials are less known. And fifthly, information on the exposure conditions is required for risk assessment. Finally, without time-resolved data, predicting actual MIE is difficult. Now, the probable solutions are: partial information from different sources must be brought together. Quantitative AOP may help in risk assessment. Omics data can provide information about all blocks of adverse outcome pathways. Availability of data for the initial response period, 1 to 3 years of nanomaterials exposition, can help most likely to predict actual MIEs. Now come to the topic. Here I am going to describe the MIE tool. Before I will mention the statistical principle behind that. Here we have developed a NanoCommons MIE gene set database, which contains gene signatures of MIs by integrating knowledge from CAGE database, Reactome database, Geo database, Wiki Pathways database, as well as custom gene sets from published data. Until date, we have collected 132 gene sets. These gene sets are responsible for 3 different types of MIs considered in this current MIE tool. The MIs are disruption of lung surfactant functionality, and second one is lysosomal destabilization, and third one is oxidation of cell membrane. So the first MIE is the disruption of lung surfactant dysfunctionality. The first question is, what do we mean by disruption of lung surfactant functionality? Here I want to mention that the alveolar lining fluid and thin layer of lung surfactant are the first protection barrier against the inhaled nanomaterials. Lung surfactant is a surface active lipoprotein complex in the alveolar space, and its main function is reducing the surface tension at the air liquid interface in the lung. Also, this complex regulates inflammatory responses. The disruption of the lung surfactant may lead to negative health outcomes, such as lung fibrosis, chronic obstructive pulmonary disease, pneumonia, asthma, acute respiratory distress syndrome, or acute lung injury, lung surfactant function disruption leading to acute inhalation toxicity. For example, the family of mesoporous carbon nanomaterials, that's MCNS, is a rising star in nanotechnology for multidisciplinary research with versatile applications in electronics, energy and gas storage, and biomedicine. Meanwhile, there is mounting concern on the environmental health risks due to the growing production of uses of MCNS. The lung is the primary site for particle inversion under environmental exposure to nanomaterials. Here, the comprehensive toxicological profile of MCNS in the lung under the scenario of moderate environmental exposure has been shown. It was found that at a low concentration of 10 microgram per ml, MCNS induced biophysical inhibition of natural pulmonary surfactant. Moreover, MCNS at similar concentration reduced viability of J774A.1 macrophages and lung epithelial A549 cells. Incubating with nature pulmonary surfactant effectively reduced the cytotoxicity of MCNS. Regarding the pro-inflammatory responses, MCNS activated macrophages in vitro and stimulated lung inflammation in mice after inhalation exposure associated with lung fibrosis. Moreover, size of MCNS played a significant role in regulating cytotoxicity and pro-inflammatory potential of this nanomaterial. In general, larger MCNS induced more pronounced cytotoxic and pro-inflammatory effects than their smaller counterparts. And second MI is lysosomal destabilization. The second one, the next level of bio-nano interactions in nanomaterials, bioadhesion to and uptake into the cell. After endocytosis, the nanomaterials by defined types of cell, macrophages, epithelial, etc., and following defined exposure routes such as inhalation, ingestion, intravenous, and dermal, eventually nanomaterials are retained inside the lysosomes. This cell compartment consists of a variety of hydrolytic enzymes like phosphatases, proteases, nucleases, etc., and a highly acidic environment providing degradation of foreign substances. Biopersistent nanomaterials can cause lysosomal dysfunction, for instance, lysosomal membrane permeability, that can induce leakage of the protease, cathepsins, and other associated lysosomal hydrolysis into the cytoplasm. It can trigger a variety of toxicological consequences, including oxidative stress, cytosolic acidification, and induced cell death pathways, including necrosis, apoptosis, and autophagy. Exposure of macrophages to the carbon nanomaterials triggers a cascade of cellular and molecular events, such as ROS generation and lysosomal damage, which serve as a mechanism underlying carbon nanomaterial induced cell death, including necrosis, apoptosis, and pyroptosis. Carbon nanomaterials cause the mitochondrial dependent apoptotic cascades through ROS activated MAX pathway, and ROS could activate several transcription factors such as NF-kappa beta that regulates the inflammatory response. Carbon nanomaterials induce lysosomal membrane permeabilization, that is LMP, resulting in the translocation of cathepsins to the cytoplasm. ROS and LMP were reciprocal causation, generating an amplification loop. LMP could potentially cause autophagy dysfunction, and inflammasome-dependent pyroptosis was initiated, characterized by cleavage of collapse 1 and downstream 1L1 beta release. And the final MIE we have considered here is the oxidation of cell membrane. Nanomaterials can induce oxidative damage of cell membranes through lipid peroxidation, in which free radicals steal electrons from the lipid components of the cell membranes. This effect might be induced by metal oxides nanomaterials. Upon nanometal exposure, ROS generation can induce oxidative DNA damage, strand breaks, protein denaturation, and lipid peroxidation, thereby demonstrating the mutagenic and carcinogenic characteristics associated with nanoparticles. Excess free radical production leads to the mitochondrial membrane damage, causing necrosis and cell death. Phagocytes includes neutrophils and macrophages generate massive ROS upon incomplete phagocytosis of nanoparticles through the NADPH oxidase enzyme system, whereas nanoparticle-induced ROS triggers an inflammatory cascade of chemokine and cytokine expression via activation of cell signaling pathways such as MAC, NF-kappa beta, AKT, and RTK. Furthermore, oxidative stress mediated stimulation of the cellular mechanisms results in transcription of genes responsible for fibrosis, EMT, and carcinogenesis. Nanoparticles elicited ROS is at the center stage for the majority of the ensuing the adverse outcomes. So now back to the description of the MIE tool again. I want to mention that we have developed a web interface with R modules of the tool. Here we have integrated MIE prediction tool into the NanoCommons knowledge base. Using R codes, we can access to the database and calculate the overrepresentation of distinct biological processes for each MIE. The tool uses a list of differentially expressed genes or proteins as input from high throughput experiments. The tool enables us to calculate a prioritized list of MIEs with identified biological processes. The services are available for transnational access. Okay. To develop this tool, we first create the databases, namely NanoCommons GS MIE responsible for 3 MIs. Already I have discussed the 3 MIs in detail. By creating MI genes using information from CAGE database, Reactome database, Geo database, HP and Wiki pathways, and public database as well as published data. The database was constructed based on the gene matrix transposed file format. That is a GMT file format. I have given the example of GMT file format here. Here we have seen that there is some entities here. First entity is name. That is indicates the name of the gene set. And second one is the description. The description actually describes what is the gene set responsible for, and among these genes, the several genes can contain several genes, and all these entities actually indicate defined gene name. This is the standard GMT file format. Yes, and this slide shows the snapshot of a small portion of the database, and the database contains actually 3 gene sets of already said disruption of lung surfactant functionality MIE, such as GEO 00431229, REAC RMMU 5683826, and SBI S001 gene sets responsible for surfactant homeostasis, surfactant metabolism, and surfactant homeostasis custom gene sets respectively. The database contains 54 gene sets of lysosomal destabilization. For example, GEO 00972124 for lysosomal membrane organization, REAC RMMU 432720 for lysosome vesicle biogenesis, HP 0004356 for abnormality of lysosomal metabolism, among others, and 75 gene sets for oxidation cell membrane. For example, GEO 0006979 for response to oxidative stress, GEO 0071451 for cellular response to superoxide, REAC RMMU 1222556 for ROS and RNS production in phagocytes, and WP 1496 for oxidative damage, among others. Now, this slide shows the snap of the MI tool web interface, and I give a link of this tool in the below of the slide. We can note down this slide link and can use this tool. And now I am going to demonstrate how the tool can be used using this link. So this is the link. I reload this link again. Yes. So this is our MI prediction tool. Here we have to set a threshold value for significance level. If you want to, here it is at 0.5, or you can set at 0.01 also. For now, I am setting that it is 0.25 threshold. You can enter your list of differential express genes, but every gene name should be in a line, and every new line corresponds to one gene name. So we have already inputted 2 example differential gene set here. I have shown you. I will show you. That is, for example, one. Suppose this gene sets already we have given to this input. We have to give input here. Here you can see that every genes in a single line. Then we have to click the run query. This is, we can see this differential express genes actually represent this gene sets corresponding to MIEs. So suppose MI2 is overrepresented for the gene sets case 04142, and the description is some lysosomal dysfunction here. And the corresponding p-value, we have getting the corresponding p-value is much less than the threshold value. So these genes have significantly overrepresented this gene set, and which genes? And this is the genes which are overrepresented by this differentially expressed genes. So we can calculate the overrepresented p-value. What is if you change this threshold value again? If you change threshold 0.01, and again run the query, we can previously we have getting 10 gene sets. Now I change the threshold to 0.01, so I getting 7 gene sets. So depending on the threshold, we are getting and the significant gene sets that were represented by your input, by your differential genes expression gene sets. So this is our example 1. If I can show this example 2 also, you can run the queries. Only 3 gene sets are overrepresented by our example 2 gene set. You can give your sample genes, your genes also. I have created a sample 1. This is my sample 1 in Notepad. Just copy it and paste here. Test. I paste it and change the threshold 0.05 and run this query. So our my new sample, I have put a new sample gene set differentially expressed genes, and I see that this differentially expressed genes overrepresent 3 gene sets corresponding to MI2, MI1 event, and 1 is for MI3 event. And this corresponding p-value is calculated, and the corresponding genes I can see here. So this is a very simple tool and simple to use. You can always go to the link of our the link you can use your differentially expressed genes and test the significant p-value for the overrepresentation. Now I will illustrate some statistical principle behind this tool. Actually, here we have used overrepresented analysis using hypergeometric distribution. In such overrepresented analysis, how the p-value of significance can be calculated. So I give an example. Let us consider there are n number of marbles, and out of them k marbles are red marbles. So obviously n minus k marbles are black marbles. 2 types of marbles here. So now we should draw small n numbers of marbles, and with the probability to find small k number of red marbles is the desired p-value of the significance. Thus the formula become what is the formula? Formula become is the success formula is p probability of finding small k red balls such that small n numbers of different marbles to be drawn, capital K numbers of red marbles, and capital N numbers of total number of marbles. Combination of capital small k out of capital K multiplied by combination of small n minus small k out of capital N minus capital K, and divided by the combination of small n out of capital N. This is the formula to find the significant p-value. Similarly, I have used this example with the marbles. Similarly, let us assume there are n number of total genes, and suppose among them small k equal to 100 genes contain transcription factor binding site, TFBS, and obviously rest of genes, that is 1000 minus 100, 900 genes has no TFBS. So we have a differential gene cluster of 17 genes, that is small n equal to 17, and among them only 5 genes have TFBS. This is a transcription factor binding site, and now we have to find whether this gene cluster overrepresents significantly the genes with TFBS or not. So according to the formula, if we put the values in this formula, we get this p-value as 0.02. According to the significance level, if we set the significant level as as a significant level is 0.05, then we can say yes, these our gene cluster of 17 genes significantly overrepresent the genes with TFBS. But if we set the significant level as 0.01, we cannot say that. So the setting the threshold level is very important to use this tool. So finally, in summary, what is the hypergeometric test? It is a probability distribution. It is a probability distribution describe the probability of genes in the it is a probability distribution that describes the probability of genes of interest being associated with a particular MIEs, and for all genes in the gene list, and form a population of all genes in entire genome which contains k number of genes associated with the MIE. And the calculation makes success follows the formula given below. I've already discussed about the formula. And finally, the resultant p-value should be adjusted for each category tested using multiple test correction. Very important thing is multiple test correction, as here we have considered a set of statistical interferences, and simultaneously multiple test problem occurs. Then we can adjust p-value using different methods like false discovery rate, Bonferroni correction method, or Benjamini, Hochberg, FTR, among others. So yes, and in our tool, we have provided an R modules also. The R modules of the tool can be utilized into various automated pipelines, including Jackpot computation platform and the NanoCommons knowledge base. Our library is a means to provide universal access to the tool and can be implemented into various automated pipelines. The ghost function, we have used a ghost function to implement the hypergeometric distribution. The ghost function from gprofiler2 library will calculate the prioritized list of MIEs with identified biological processes. So, behind this tool, we have used the function ghost function. We have to install. If you use the R program, we have to install the package gprofiler2. How to install this package gprofiler2? Here, this is the code. Sorry, install the packages gprofiler2. If we use the Anaconda platform, we have to use this code to install this gprofiler conda install -c conda -forge or -gprofiler2. Then we have to use the library gprofiler2 and perform this function. Here we have seen that it is performed this function ghost. Ghost is function actually do the same thing we have seen in the hypergeometric distribution. Just we have here only is that we have here if we ordered query one argument here ordered query. If we put ordered query as a false, it becomes a simple hypergeometric distribution test. And if we put ordered query as true, it becomes a incremental enrichment analysis. So if we put ordered query as true to make hypergeometric test to perform the incremental enrichment analysis to find the key MIEs event gene set for your differential express gene input. So here ghost function has the argument query. Query is nothing but your differential express gene. I will demonstrate that. Organism is the organism that is we have used 3 MIEs, MI1, MI2, MI3. So we have already created a predefined token. For this predefined token contains the all 132 genes we have provided already in our database. This predefined token contains the 3 MIEs gene set. So you do not bother about the if we want to use the three, want to analysis about these three MIEs, we have to put this predefined token for that. That is okay. For that will be okay, and you will not bother about the background. And domain scope of, okay, obviously we have put the known correction method. A special correction method already I have considered the FTR method is one multiple correction method. GSC is another type of correction method we have used from gprofiler, and it is developed version of the FTR multiple correction. And we have put the user threshold p-value. It should be 0.05 or 0.01. You can put here, and this F code should be true. And ordered query true, ordered query true, it will become the incremental enrichment analysis. If it falls, is simple hypergeometric test. So instead of GSCS, you can use also FDR. You can use on Bonferredi corrections, Benjamini, Hodgepap, FDR. If you want, you can use. You have no problem. I have just seen how the in R you can run this ghost function. This is the I have seen. This is the only R studio, and we have write this same code here. I've already installed gprofiler 2 using pip just command, just install the packages gprofiler 2. I have already installed these packages. I have used the library gprofiler 2. I have set p-value as 0.05. So I have a file and named differential dig list import implement 2.txt. Here this file. Yes, this file has some 1, 2, 3, 4, 5, 6, 7, 8 differential expressed genes. So I have just read this file using read telim. This separated by new line. I already said we have to give input genes in a new line. Every gene should be in a new line. I have put in genes in a new line, and this is already, when I read this file, it becomes a list called a list data structure of the R, and we have to put this vector out input to our ghost function. So I have convert the list using unlist command to a vector and store in query 1. And this ghost function, similar ghost function I have written in the slide. The ghost function query 1 actually just run only this part. This I run this part. Yes. Now in query 1, this is the query 1. You can see the query. Sorry. Query 1. Oh, sorry, the name is wrong. QERI. Yes, you have seen this same genes. I have read the same gene set. This is this same gene in a vector format. Okay, and this gene set, FS gene set, I put as the input in query 1. Now organize, I put this predefined token, and correction method I have used FDR here, not GSCS. I can put also GSS also. User threshold p-value, I will already set the p-value as 0.05, and ordered queries true. You can make it also false also for only hypergeometric test, and if you make it true, it becomes incremental enrichment analysis. So I run this function, run ghost, and result would be stored in the run ghost result ghost variable, and I run this code. Yes, yes, the is run, and we see these three MIEs. MI1, MI1, three event of MI1, and 3 gene sets are overrepresented by these differential expressed genes. Are corresponding p-value is this one, very small, e to the power minus 21, e to the minus 0.08, e to the minus 0.03, so very small. So if you put the threshold value as 0.0 or no matter, so this 3 gene sets are significantly overrepresented by our given sets of differential genes. So you can use our tool using ghost function also. So if you write a code for your big project, you can use the ghost function. And if you want, you can use our MIE prediction tool, because you have nothing to worry about writing a code. You have put this your differential expression here and put your threshold value and click the run query. You can see the result. Okay, and you can also download the result also. So you can know worry about writing the code ghost function, etc., etc. What is the argument? Just put your differential expression on line by line and run the queries and get the output and download the result. That's it. So now I'm trying to concluding my session. So the slide. Thus, the entire pipeline can be described one after another. And for first one, I have to create an MIE gene set database. We have already create this database. Then we used the R. You can use the Python code also, but we have add stress on the R code. Client library is gprofiler tool to perform an over-representation analysis for MIE terms using cumulative hypergeometric test. And the from user end, they need to input a list of genes or proteins and press the run query button, whatever I have told just sometime before. Here some key points should be mentioned. The genes can be derived from omics experimental data such as RNA-seq, microarrays, mass spectrometry datasets, among others. The tool performs, already I've said, incremental enrichment analysis, and this list of genes is interpreted as an ordered list where elements are in the order of decreasing importance, and the order of genes can represent some biological effects, obviously. And finally, the web interface accepts a list of genes, one genes per line, in defined formats, gene symbols format, it can be gene IDs format, etc. Mixed types also can be entered. No problem. And in conclusion, I can say that the tool can fill the gap in understanding of bio-nano interactions and facilitate reconstruction of the sequential chain of key events in a particular adverse outcome pathway. In this first version of the tool, we integrated gene expression predictors for 3 key types of MIEs: disruption of long surfactant functionality, lysosomal destabilization, and oxidation of cell membrane. And these MIEs were highlighted and described as the initial starting point of toxicological responses to nanomaterials for pulmonary adverse outcome pathways in the Smart Nano Toolbox project. And the tool will be extended and updated during the NanoCommons project to address the needs of nanosafety community. With this deliverable representing the first such tool to be integrated into the NanoCommons knowledge base. Thank you for your patience.
Thank you, Abhijit, for your nice overview on the background of the tool, as well as showing us insight into the use of the tool, even going into the statistical background of it. I have seen during your talk that there were 2 questions basically coming up so far. I would still like to read them. Actually, to a certain extent, you have answered, I guess, on your final slide questions already, but let's reiterate. One was actually how relevant these 3 molecular initiating events and the associated gene sets for the environmental species. So maybe you want to quickly reiterate on that. It's
I can
confined for the human pulmonary tract,
yeah,
right?
Can you hear me?
Last slide, I have answered the questions in the last slide, the toxicological responses. This is MIE highlighted and described as the initial starting point of proximity responses to nanomaterials for pulmonary adverse outcome pathways in the smart nanotox project. So I've traced on the pulmonary adverse outcome pathways initially, and if we need further, we added new MIEs for in this tool as per the need of the ongoing project.
Yeah, regarding another type of organism, currently we developed for mice, and actually we can transform genes to human or rats. But regarding environmental species, not sure that applicable. Currently, I think that definitely we can use this data for in vivo for human mice and rats. Experimental data, and for in vitro experimentals, like for example, work with macrophage or epithelial cells, and we can analyze molecular initiation events using these models.
Yeah, the second question was going into a kind of similar direction, maybe, but still we could shortly discuss it. It was actually, is the web interface for predicting the molecular initiating event from gene expression designed specifically for inhalation toxicology? So, I mean, I have seen that lysosomal disruption, for example, this might be a relevant point also for injected nanomedicines, for example. Is this somehow on attention?
First of all, we developed tools specifically for lung organ, and actually it's I suppose in terms of in vivo experimental data, it's inhalation, of course, experimental data. Yeah, but also we can use this for in vitro data. I suppose it is impossible to use for another type of organs, just because this is a molecular initiating event, just specifically developed for inhalation.
Okay, thank you. And there is one more question I see in the chat. Maybe I missed the information at the beginning of the talk. Was this database constructed only by these 3 molecular initiating events, or are there any other molecular initiating events implemented? Well, at present not. Maybe you want to say a little bit on what is in the pipeline. Are there any things more planned regard to?
Currently, we developed 3 molecular initiating events, but actually we can use the same scheme for extending to extending the number of molecular initiating events, and technically it's feasible. Yeah, but currently we just develop only for 3 molecular initiating events.
Another one, when the sample is taken for molecular initiating events, lung surfactant disruption, if a substance disrupts function, the lungs will compensate for the disruption, so the timing will be important. How about timing? Possibility of linking different time points together?
Yeah, thank you, Jared, for this. Good question. Its timing is really quite important in this case, and currently we just used published data during, for example, after Smart NanoTalks project or another project, and mainly we focus on first day 1 or at least day 3, and unfortunately we don't have actually time series experimental data, and it's really important maybe just to take to account in the future project, just to analyze different time points and what's happened during after 1 hour of inhalation, 2, 3, and so on, especially for molecular initiating events, because this is initial step for interacting nanomaterials with cells. And unfortunately, we don't have a lot of data for analysis. We have some limitation in terms of time points and so on. And that's why I would say that it's some kind of preliminary data, or just preliminary results, or just first step in this direction.
One more question. Are there plans to expand the list of MIEs for inhalation toxicology and eventually order for other organ systems?
Yes, we are going to expand to humans and rats, and technically it's feasible. We are going to convert gene names to one of the organ, one of the organism. Regarding other organ systems, not yet. We don't have plans for expanding to other organs. We currently just focus on only inhalation studies. But actually, if you will have just really good data set with time points, and actually we can analyze this and we can expand this tool to another organs.
One more question was, what is the domain of applicability of this domain in terms of particles, chemicals, different physicochemical properties or others?
I'm not sure. Maybe I didn't catch what does it mean domain, but I suppose it can be maybe some type of foreign materials. And actually, we mainly focus on nanoparticles. It can be, I don't know, carbon nanotubes or titanium oxide or carbon black and so on.
So maybe I ask one thing which I was interested in. Actually, the establishment of the molecular initiating events was done based on carbon nanotubes for the first two, and on metal oxides for the third molecular initiating event. Is this correct?
Actually, we try to develop some kind of universal tool without focusing on specific nanoparticles for molecular initiating 1, 2, and 3. And I suppose that we can, all three molecular initiating events can be applicable in case with carbon nanotubes, carbon black, or other or any physical nanoparticles or any nanoparticles, any forms or any chemical structures and so on, because it can be different type of interaction between nanoparticles and cells. And it can be, for example, if we will use the long nanoparticles like fibers, it can lead to frustrated phagocytosis, which can lead to generation of reactive species and so on. All some nanoparticles maybe can interact directly with the surface of cells and induce lipid oxidation and so on. Or, for example, hypothetically, for example, maybe some nanoparticles which form some conglomerate with proteins can actually interact with one of the receptors into the surface of cell, and also can interact. There are a lot of different potential type of interaction between nanoparticles and cells, and it can be maybe direct interaction, like lipid oxidation, for example, or indirect, when macrophages just generate reactive oxygen species, and this reactive oxygen species can disrupt cell functionality or cell membrane in epithelial, for example, cells. Yeah.
Okay, thanks a lot. Maybe one word regarding experimental validation. we suppose that all these omics-based data were kind of also experimentally validated by other kind of specific experiments. So these data sets are very validated ones.
Yes, it would be really good if we can validate molecular initiating events, because currently we can use microarrays data or RNA-seq data, and we can see how we can check alteration of gene expression or mRNA level, for example, of before inhalation, after inhalation. But actually, maybe interesting just to introduce orthogonal experimental data during maybe just another type of experimental experiments and instruments, and just specifically focusing on molecular initiating events or how nanoparticles can interact with cells.
Thanks a lot. So we had Vadim Tsernovkov, who was answering most of the questions. Thanks a lot, Vadim. You were deeply involved in the development of all this, what Abhijit did so nicely present to us today. So thanks a lot, both of you. Thanks a lot also, Vladimir, for the project actually on which this was based on, the Smart Nano Talks. I would like to remind at this stage all the participants that all the materials, the slides, as well as the entire recording will be available online, either via the NanoCommons infrastructure, you can access it, or you would find it on the Elixir tests on the NanoCommons channel there, or the NanoCommons channel at YouTube. So you find a nice collection of these topics. Today we had a look at the molecular initiating events that are guiding into adverse outcome pathways in modern theme of understanding toxicology at the very mechanistic level, here even at the molecular mechanistic level. Thanks for all the presenters. Thanks for the audience. Thanks for being with us. Stay tuned with us. Stay tuned with the working group A of the Nano Safety Cluster. We are having regular webinars and more interactive trainings in near future. Consult eventually also the Nano School offered the Venice Nano School. I have shown the web link there, which is going to touch upon different topics of the entire nano safety data lifecycle. Thanks very much. Have a nice afternoon or morning or even evening, wherever you are beginning in from.